right? Well, if we know the energy of an intermolecular force, why can't we go through and then predict what interactions we would get out of it? Okay? Why can't we predict the actual three-dimensional structure? So number one, we can. Number two, it takes a lot of calculations. Remember, if we had 300 individual hydrogens and how each of those hydrogens could potentially interact with each other, we have to calculate the energy highs and the energy lows and try and put that together to come up with an overall structure. Okay? Not an easy process, but we do have the ability to go through and do that, and hopefully, I think going full screen is a bad idea, but let's see. So what they're showing in this process, or this video, is someone actually went through with a small protein, and they said, okay, let's predict the three-dimensional structure. So they took their protein, they have it highly denatured. Denatured meant uncoiled. So we removed pretty much all of the three-dimensional characteristics of the model. We have pretty much just a primary structure, okay, just the sequence of amino acids. They went through and said, okay, here are all the possible interactions that can occur in the backbone of the protein, and said, okay, now that we know what all those possible interactions are, we know the highs and the lows, let's go through and try to mimic and predict the three-dimensional structure. So what is happening in this case now is they're letting their computer model predict it. So it is moving each and every single one of those atoms or bonds in a location so that they can see how it would interact. You notice almost right away, what do we end up seeing? We've got an alpha helix almost immediately forming. Okay? Why? Alpha helices are very, very stable. Okay? So if there is the potential for an alpha helix to form, it's an energy minimum. So we almost always will see that form right away. And then the rest of them are a little bit harder to see. Let's see if I can get one. Oh my gosh, it's like trying to pause. This has got to be going double speed. This is ridiculous. Okay. God, it's going way too fast. Oh, there it is. No, come back here. I see you. Of course, now the arrows are gone. Did everybody see the arrows in the back? Yeah. What was happening there? Okay, we're getting a beta sheet. Deciding anti-parallel or parallel is a little bit trickier because we'd have to see how it would loop. Oh, good point. It did have the arrows. And I'm not going to be able to pause it. So if we timed those arrows, I think you were right. This has to be going extra fast. This is ridiculous. The more important aspect out of this that I think is kind of fascinating is that they went through, predicted what they expected out of this. Okay. And so this is now the end result. They ran the model several hundred times, and then overlaid the end results out of each of those models, and they ended up with something like this. So they're just showing that each of the model systems roughly came out to something pretty close. Okay? Each time they had some slight variance within it. Okay? What I think is a little bit more interesting, right? Now we've got the arrows, is they went through and came up with this overall structure. Okay? And if you read that text, this is the best obtained structure of crystallographic quality superimposed on the native. What does that mean? They took their model,
superimposed it on the actual,
and they superimposed it on the actual crystal structure. So based off of spectroscopic data, they were able to determine the structure, and they superimposed their model over the top. Look at how freaking close that is. Okay? Pretty amazing, just based off of understanding how the intermolecular forces, admittedly a bunch of other stuff as well, overlay and interact with each other. They can use a computer model to predict the three-dimensional structure starting from a primary sequence of amino acids. Okay? Absolutely fascinating. Okay. So, back to dealing with this. That's about all we get out of it. You're not going to be dealing with the computer modeling. You won't have to do the NMR. You won't have to do the X-ray crystallography. You're just responsible for knowing that these tools are out there that we can go through and do this. The part that we kind of cut out of Chapter 4, at least for the exam, was referencing myoglobin and hemoglobin. Okay? These are both insanely important proteins, and as such, you have a new homework assignment, which was originally folded in, where it's just the hemoglobin and myoglobin questions. You might need to go out and reference, say, Wikipedia about those individual proteins to help you kind of answer those questions. It's important enough that you read about them, because I know hemoglobin and myoglobin show up again on lots of standardized tests. Okay? So that's the primary reason those questions are there, is to kind of address some of these things so that you can do better on future tests. So myoglobin's interesting enough because it was the first protein that they discovered via X-ray crystallography. Okay? So this is kind of neat. 153 amino acids. Okay? Each amino acid, you got at least 3 carbons, a couple oxygens, nitrogen. You're looking at a pretty massive structure, even with 153 amino acids. Okay? Not only is it just the amino acids, but we also have a heme. Okay? The heme is what they're showing, that little red group in the middle there. So the protein folds around the heme. Okay? It's also referred to as a prosthetic group because it's not part of the amino acid sequence, but it is something necessary for the overall protein to function. Okay? So the protein will fold very, very tightly around the heme, and the heme holds on to an iron atom. This becomes important because myoglobin is used as to carry oxygen. The heme will bind oxygen. Okay? So our protein binds the heme binds oxygen, and we now have a way to transport oxygen from one site to another using this globular protein. Okay? Why use the protein at all? So that's an interesting dilemma. If the heme binds oxygen, why do we need the protein at all to transfer oxygen?
Transport around.
The heme can move just as easily as the protein. In fact, couldn't we make the argument that the heme is going to move a lot easier than the protein? The heme, if we look at its size relative to the protein.
Is it similar to hormones, and that the protein prevents it from being excreted through the kidneys?
Okay, so the protein could make sure that the heme stays in our bloodstream. Okay, that could be valuable. And in that whole excretion process, we've designed a system to excrete things. Okay? If heme worked, we wouldn't excrete it.
Maybe regulate the uptake and release of the oxygen according to the different pHs of blood, because the protein is going to change shape and allow for oxygen to be released or be taken out depending on the shape of the protein.
So the really important aspect that we're looking at within this system is that the heme not only needs to be able to bind oxygen, but it also needs to release it. Okay. The heme structure on its own is relatively tiny and compact and won't change its shape, which then means oxygen binds, and then it stays there. So that's great. We could then move a heme with oxygen to it anywhere we wanted, but then the oxygen is stuck to the structure. The whole point is the oxygen needs to come off and interact with whatever needs to use it, okay, through cellular respiration. So the rest of the protein structure is actually there to disrupt the ability of the heme to bind oxygen. That way, we can then kick the oxygen on and off of the structure. The easiest way to regulate kind of an on-off switch, as you've already suggested, is looking at the concentration of hydrogen ion. What happens if the pH is really, really high? It's basic, which would mean any acidic functional group would be deprotonated. We now have a charged state. That charged state is going to have a drastically different interaction than if we were under an extremely low pH or acidic conditions, and the protein structure will change. That protein structure changes just enough that it will affect how the heme binds oxygen. So it is highly dependent on pH. So we didn't talk about what the hydrogen does. Right? That is an important aspect. Does the hydrogen cause it to bind oxygen, or does the hydrogen removal of the hydrogen cause it to release oxygen? We didn't address that, at least not yet. Kind of a blow up of the heme so we can get a better idea of what's going on with it. The heme, for the most part, is a flat molecule, which is what you can kind of see in this image. They're sort of attempting to show that flat, but it's overall flat with the iron coordinated to the nitrogens of the heme group. Oxygen would then bind in kind of an end-on fashion, perpendicular to the plane of the molecule, okay, either from the top or from the bottom of that iron atom. The rest of the protein structure kind of comes in and hides part of the iron so that the oxygen does not form a perfect coordination to the iron. It kind of comes in sideways, okay, or a little bit tilted. This facilitates a better removal of our oxygen. Before we completely, well, we'll come back to it in a second. The next section when we looked at protein structure was moving up to quaternary structure. So myoglobin is our standard tertiary example because it's just the protein holding onto the heme. It folds, we now have a tertiary protein. When we move up in complexity, we can move up to the quaternary structure, and a classic quaternary structure is then hemoglobin. Well, what's the difference? Hemoglobin is shown in our lower left-hand corner. There's 4 units. Okay? It's not myoglobin, but it's the same kind of idea. If we looked at a single hemoglobin protein, it looks very, very similar to myoglobin. And well, why might that be true? What does hemoglobin do? Same thing as myoglobin does. So the structures are very related. Okay? Something has to be pretty drastically different. That something is because the hemoglobin proteins, their tertiary structure, interact with other hemoglobins to form a tetramer, where 4 different proteins all come together and interact. This becomes important in addressing this other topic of an allosteric effect. All that's happening when we mention an allosteric effect is that the binding of something causes the structure to change in either a way that facilitates more binding or less binding. Okay, so allosteric is really just saying changing structure. Okay. Hemoglobin is our classic example for allosteric positive cooperativity, meaning that when one oxygen is bound to one of the heme groups, that causes the protein structure to change. In that individual subunit, when it changes, it causes a change in the other proteins around it, which then allows them to bind oxygen better, which causes a further change, which causes the next protein sequence or protein structure to alter, which then allows it to bind oxygen even more so, and it kind of continues to go around. So hemoglobin has this structural effect in how it binds oxygen. Okay? The more oxygen, the better it binds oxygen molecules. The less oxygen, the less likely it binds oxygen. Okay? So we looked at this. This is now starting to look at back with our acid concentrations. Remember we asked this question, what happens to our pH? And we looked at blood buffering. Okay, we've got carbonic acid and CO2. What would we expect to happen to the pH in our lungs? What do we want to happen in our lungs? We want to get rid of CO2. So to have CO2 leave, what needs to happen? To shift the equilibrium in that direction. How can I shift the equilibrium in that direction? If I can increase the acid concentration, that's going to cause a shift to cause CO2 to be released. So we might expect, just based off of the buffering system, that what would our pH be in our lungs? Relatively low. It should be fairly acidic. And in our muscles, we would expect the opposite. Because in the muscles, we want the blood to pick up the CO2. Okay? But is that true? No. If we think about when we do physical activity, what happens? We feel that burning in our legs, I do more running than anything else. General muscles, whatever you're working with, will get kind of sore and feel a burn associated with it. What is that burn?
Lactic acid.
It's an acid. Okay? The pH in our muscles tends to be lower than the pH in our lungs, which is opposite this buffering system. That seems a bit weird.
But the concentrations are different in your muscles and in your lungs
But that's of
oxygen and carbon dioxide.
true, but if we look at our buffering system, we would want to increase the pH to kick out CO2. We're just looking at the buffer. Okay. So this does seem a little bit counterintuitive to what we would already have expected our bodies to do, and that's a bit weird. Okay. So what's going on with it? Well, if we go through and actually look at hemoglobin and myoglobin, are they only binding oxygen and CO2, or are they only binding oxygen? Let's just stick with that.
No.
No, what else are they binding?
CO2.
CO2 and hydrogen. When we're looking at our blood buffering system, okay, when we try to decide where would we expect the pH of our individual locations to be, all we were looking at was the buffering system. Okay. It turns out hemoglobin and myoglobin function like a second buffering system, which then means we see kind of a change that matches what we would expect within our own physical bodies. Okay? So it's not quite as simple as just looking at the buffering system. The buffering system isn't responsible for the kick out of CO2. The buffering system is responsible for maintaining the pH. Okay? The hemoglobin or the myoglobin is the one that's responsible for the release or absorption of oxygen and CO2.
That's why the buffering the whole system is what keeps the oxygen on the hemoglobin until it gets where it needs to go.
Yep.
Otherwise it would dump out too early.
Yep. And it's also going to be dependent on the pH as well, because as we alter the pH slightly, that's going to change the structure of the hemoglobin. If the hemoglobin structure changes, what happens? It may release the oxygen or not. So if we go through and actually take a look at our reaction now, okay, for our hemoglobin, what happens when we are at low pH? So low pH means what happened to the H+. It's really high. Okay. At a high pH, or sorry, a high H+ concentration, what happens? It shifts to the right, and we release oxygen. When your muscles are burning, what's that mean?
You need more oxygen.
We need more oxygen so that we can actually effectively produce energy. Okay, so what happens? Hemoglobin in the bloodstream will notice the high acid concentration, and what does it do? It releases oxygen. At the same time as it releases the oxygen molecule, its affinity for CO2 increases, and our muscles can help start to relieve some of that CO2 buildup. Okay. What happens if we're at low pHs, or sorry, high pHs? What does that mean?
We don't need the oxygen, we can take it up in the hemoglobin.
So, first step first, a high pH means low H+, which means which way does our equilibrium favor? It'll go the other direction, which means what?
It's going to take up the oxygen.
It'll take up oxygen and it will release CO2. Okay. So when we're looking at our blood buffering system, the blood is really there, or our buffering system is trying to maintain the pH as best as possible. Okay. We will do slight variations off of that, because that slight variation will cause our second pseudo buffer, our hemoglobin, or myoglobin for that matter, to respond to that subtle change in the acid concentration and either bind oxygen or release oxygen depending on where we're located. So what does that mean? pH is in our lungs. We would
average is like 7.4. It's on the higher end of that.
It should be on the higher end,
Higher pH.
a higher pH, because we have less acid present, okay, and that will facilitate the binding of more oxygen. Whereas in our blood, we would expect
A low pH because you want to release the
a
oxygen.
low pH because we want to be able to release the oxygen to our muscles. Okay. So when we evaluate and try and predict how different things interact with each other, it can't be as simple as just one thing. We've got to realize there's a bunch of different aspects all tying to each other. Every protein will be affected by the acid concentration, which then means we're really looking at two buffering systems when we go through to predict our reactions. Okay. So, hemoglobin, H plus CO2, very key aspects. We also have this molecule BPG. Okay. Why do we want three different things affecting oxygen binding?
Because sometimes we're in distress and we need to.
If we encounter a different stressful environment, we want to be able to change relatively quickly our response to either the uptake or release of oxygen. BPG helps to contribute to that. Okay? So we need a lot of that dependence so that we can control that relatively well and with a variety of different tools. Because if we lose access to one of those abilities, we need some redundancy in the system so that we can still maintain a functioning system. Next thing we'll look at is an interesting graph here, percent saturation versus oxygen pressure. Right. We're going to be looking at lots of graphs in the coming sections. So we need to be able to interpret what's going on here. Okay. You'll notice myoglobin spikes upwards very, very quickly and then kind of levels off. And the hemoglobin's kind of flat and then spikes and then levels off. Why do they level off? Fully saturated. We can't put any more oxygen into those individual molecules. That's why they're leveling off. Okay. Why is myoglobin spiking but hemoglobin not?
Because hemoglobin has 4 globins and myoglobin has 1.
It's also affected by the amount of oxygen on it.
There's our issue. It comes back to that allosteric interaction. Okay. If they had the same binding affinity, hemoglobin should be able to pick up that oxygen just as quickly. But it's level at first. Why is it level at first? This is that allosteric effect. That first oxygen binds relatively weakly. Okay? It doesn't associate very well. As the oxygen binds, what now starts to happen? We increase the affinity for more oxygen, and now we see a spike. Okay?
That's like working the other way too, so it's hard to get that first or the fourth one off, or the fourth one to off pretty easy.
It would be harder to get that fourth one off, yeah. Okay. So what we're noticing is that sigmoidal curve for our hemoglobin. Sigmoidal is characteristic for an allosteric interaction. So if we looked at the saturation, we would see a sigmoidal curve. Okay. If it's not allosteric, it binds almost instantly and it levels off almost right away. Why? Because there's only one site. Once it's hit there, it's filled, we're done. Okay. There's nothing for it to affect or change the structure of. Okay. Questions? BPG is another floating thing in the blood. And I know I totally have a slide that discusses BPG, and I'm pretty sure I did not see that show up here. Dang it. Why did that not make it in? Your textbook addresses BPG in reference to fetal versus mother blood. because what we're looking at is the ability to transfer oxygen from the mother, which has lungs, to a baby, which does not. We need to be able to facilitate that transfer of oxygen. Okay? So we end up seeing a change in the concentration of myoglobin versus hemoglobin across that system because we need to be able to get the oxygen across to the baby. So if we were looking at a baby, which one would we expect to see a higher concentration of? Myoglobin, because it needs to steal the oxygen from the mother. Okay? The mother's the only one supplying oxygen. The baby needs to be able to steal it from the mother to survive. Okay?
But do adults also have?
We do also have it. We start to not need it as much because we're not relying on something else, somebody else to breathe for us. Okay. Chapter 5 is literally all about protein lab stuff. Okay, so if this was our protein, what could we take advantage of to isolate it away from other protein or amino acids? How could we isolate this amino acid from a bunch of others? If we change the acid or base, or we change our pH, that'll change the charge on the structure. If the charge is different, what could we potentially do? Yeah, we've already protonated or deprotonated it. We can now try and separate. How do we separate? See if anybody remembers. Zwitter ion just means that there's two charges that's charged, so that doesn't have to do with this at this point. Gel. Electrophoresis. What is electrophoresis?
Stuff in the DNA in the gel, and you positive and negative.
We're putting charges on either ends of a gel, and what's going to happen is, depending on the structure, it's going to be attracted to one end or the other because of charge. So we can separate and isolate varying amino acids, and for that matter proteins, by varying the pH.
If you vary the pH, I mean, wouldn't that affect all the molecules that are in
Yep.
it? So potentially you could have a lot of them that have the same charge?
Yep.
Because they're-
So you could end up with a lot that have the same charge, but you will have now removed some of them. Once you've now got a smaller batch and a slightly more pure system, you then have to switch your pH and change it again to try and continue to purify.
That goes back to each amino acid has its own pH thumbprint almost, and that's what.
Yep. So you'd be looking at PIs, and when we go through to separate, can we separate only based on charge?
Size.
We could separate based on size. How? That's what your gel is ultimately doing, is separating based on size. Okay? The larger it is, or the more polar it is, the slower it'll move. So we can separate our amino acids based off of polarity as well. At least I'm not the only one feeling like crap. Okay? That's all we pretty much want to get out of chapter 5, so we'll move into chapter 6. Okay? Chapter 6 is dicey. Okay? Everybody's going to have a problem with this chapter. Okay? Even I have a problem with this chapter. So we'll take it kind of slow. If you've got questions, ask them. Okay? So we're going to look at kind of a brief separation on this. We've already kind of talked about thermodynamics, but we kind of left it alone. We're now looking at kinetics. Okay? We want to know how fast an individual enzyme works. Once we know how fast it works, we can start to predict something about how it functions, what it interacts with, and all those other processes. Okay? So since we're looking at a biological catalyst, in our general energy diagram, what would that mean? If I add a catalyst, what would happen?
Kinetic energy in the transition state would be lower.
We would lower our activation energy by changing the mechanism, changing how that reaction works. All of your biological catalysts function this way. Okay? We change the mechanism, which is why we end up seeing potentially two humps. Okay? We have a new intermediate, and that drops our activation barrier. Okay?
What is the second one?
We've changed the mechanism, which means we potentially end up with an intermediate. Okay? So that's why we have that next valley, is a new structure is formed. Once we have that new structure, it can transition to the products easier by going over a second reaction.
The enzyme changes the original structure, and then the
Yep.
original structure still does what it was going to do.
Still does what it needs to be able to do. Yep. And this can be done as simple as adding a hydrogen or removing a hydrogen. Okay? That can change your mechanism enough that it now becomes relatively easy to initiate. Okay. So in our qualitative discussion, we've got two primary mechanisms, or two primary, let's stick with mechanisms, models for how an enzyme works. We have the lock-in key and the induced fit. You guys familiar with those terms? Only
The
saying
whole lock and key
sort of.
thing, like that recent research is saying that that's not its best description or something.
Yeah, it doesn't work very well. So the idea on the lock and key is it's very static. Okay? Think of the lock and key on a door. You stick the key into the lock, and if the key matches the lock, you can then twist the key and you unlock the door. Same thing would be happening with the enzyme. The substrate binds perfectly to the enzyme, right, and it will bind in a place known as the active site, because that's where the chemistry is going to occur. It forms a new complex known as the enzyme substrate complex, because they're now connected to each other, very original name. Okay. The chemistry can then occur, and it can then spit out the individual products that come out of this. And that is not actually shown on this diagram. But we could add an arrow, and we'd end up back with our enzyme, and we'd end up with our product, whatever our product was.
The way that I'm kind of told is that the enzyme binds and changes the shape of the original molecule, which then...
So in this system, we're saying that everything fits perfectly, like a lock and a key. The key, or the lock, doesn't change its shape when the key comes in. Okay? That system or that model is very outdated and doesn't make a whole lot of sense. Most enzymes are going to function under something known better as the induced fit. And what does that mean? The substrate and the active site within the enzyme are kind of close in structure, but not perfect. So what ends up happening is the substrate comes in, interacts with the free enzyme, and forms the enzyme substrate complex where they now bind tighter to each other. That change in structure can facilitate the formation of our product. So we end up getting the same overall result, but through a slightly different understanding of the individual aspects of it. Okay.
How the atoms interact with each other.
Yep.
Manipulates the shape.
Changes the structure. And that makes sense. When we changed just by adding or removing a hydrogen from a single structure, we got massive overall changes in the physical properties of that molecule. We're now binding an entire second molecule. We would expect some significant changes in your enzyme structure. Okay? So the lock-in key really is not a very good model. Okay? It's a nice one that works for, say, 5th graders. Not so well in the real world. Is there a question there?
So if, I thought that the enzymes pretty much there's no change to the enzyme going through the process, right?
So what do you mean by there's no change?
Well, I thought that, an enzyme, it attaches, it does what it's gonna do, and then it separates, and it is the same, it's in the same state that it was when it attached.
Yes.
So it seems like if it's changing, something's making a change to substrate, you would think there'd be some kind of transfer of an atom or something that, or you would just think once it separates that the substrate would just go back to the shape it was in or something, or I don't know.
So what the enzyme does when it grabs that substrate is it is for sure changing structural effects within the substrate.
Okay.
And at the same time, it's going to have to facilitate bond transfers. And so when we get to this enzyme substrate complex, the enzyme loses its enzymeness, if you will, because it's no longer the same. It has now changed its structure. Okay? At that point, you are correct. We're no longer looking at a catalyst. But if we looked at any other kind of catalyst, say a reaction of an alkene with platinum and hydrogen, okay, that overall reaction put hydrogens on, right? But if we look at the mechanism behind that, what's our... shoot, I didn't want to do that. what does it actually look like? Well, we've got our platinum surface that binds hydrogen, and it binds the carbon structure. At this point, do I have just platinum? No, I have this big complex structure with hydrogen and carbon bound to it. So in your argument, then we don't have the enzyme anymore. Same thing's happening here. We don't have the catalyst. But what happens is once we've now formed that complex, the enzyme facilitates, or catalyst facilitates the transfer of electrons, transfer of those bonds, and ends up spitting out the final products. And reforms the platinum surface all by itself. The same thing's going to happen with the induced fit model. The substrate binds, we form this new intermediate, this new complex. Stuff happens, it then falls apart. And in that process of falling apart, what we end up forming is now a product, or products, as is more likely the case. And we'd reform our starting enzyme.
Doesn't the enzyme need to, I guess in this case, pick up some hydrogens somewhere to get back to where it was? Or?
So that's a large part of how the individual reaction works. Okay? How does it facilitate those hydrogen transfers? Okay? Is it coming from one molecule? Is it coming from multiple molecules? That depends on what the enzyme's actually trying to do. Okay? The end result is that you will still always make it back to the starting enzyme condition, even with those structural changes. It will go through and completely reset at the end of its full loop.
Could an enzyme also work with like hydrogen bonding, so it's not transferring anything, it just does its thing, everything but it's part of it.
The hydrogen bonding is a massive, important interaction that's happening in that active site to get the substrate to come and actually be attracted. To facilitate whatever reaction is occurring, which we'll see when we get into, depending on how we go, I think we'll go through glycolysis. We'll look at what's happening in that active site, and we'll find that we are transferring atoms. We have to transfer atoms. So there is some kind of bond transfer, but in that process, that is really just your intermediate. Okay. It facilitates all those transferings of electrons, changing the structure of the enzyme, changing the structure of the substrate. Okay. But it has to go through a sequence of steps. Once it's finished those sequence of steps, it will spit out the product, and the enzyme will have reverted back to its initial state. Okay. Most cases, all of these structural changes could happen theoretically on their own within the substrate. We end up forming too high of an intermediate, so it can't do it by itself. The enzyme comes in and drops that transition state by changing how those atoms interact with each other. So what you could potentially envision is that your substrate just straight up cleaves, could cleave on its own. That is too high of an energy, so it can't do it. It binds to the enzyme says, okay, here's now where you can cut. Now you can break apart. And then it pops back out as they now broken apart pieces, and we're left right back with the enzyme, like a pair of scissors.
But the enzyme minus the hydrogens or whatever it would do.
But remember, the only hydrogens are coming from the substrate. Everything that you need to do the reaction is already in the substrate. All the atoms.
Okay, so it's not then like the platinum hydrogen you just showed.
When we look at the platinum hydrogen and the carbon structure are the substrate.
Oh, because we added the H2, so the H is on the platinum, which is coming
Okay, so
from the
the
H2.
platinum's the enzyme.
The platinum is your enzyme,
Carbon are the two-
hydrogen and the carbon are your substrate. Okay. This one's a little bit more complex in the organic example because we're looking at two substrates. Okay. Could we look at a system like that? Absolutely. Okay. Hemoglobin is a potential example of that. Once one oxygen binds, it affects the binding of another oxygen. It's the same general idea.
So the enzyme is speeding up or lowering the energy versus substrate to do whatever it's going to do.
Yep. Exactly right.
But the binding of the oxygen wasn't part of the substrate or the enzyme, it was in the system in which it's bound. I
Yes.
think we have to remember that part. So the hydrogens don't necessarily have to be on the platinum or on the substrate, they can be in the environment. and
They
the
just have to
enzyme
be in the
grabs
environment.
Those
Exactly right.
hydrogens and puts it onto the substrate, and it
Yep.
doesn't change the enzyme.
Yep. It doesn't change the enzyme at the end. Okay, and that's an important distinction. A lot of stuff can happen through the middle. Okay, for it to function as an enzyme or a catalyst, the beginning and the end state must be the same. All sorts of junk can happen through the middle. We can then push that a little bit further, and this is where we're seeing now the product happening within it. So they're saying once we've formed the enzyme substrate, okay, it facilitates some chemistry. Okay? In this case, they're saying a piece now disappears. That's a lovely way to describe this. And then that product now doesn't have the same affinity for the enzyme, and so what happens? It releases the product out into the solution. What was that piece? More than likely, okay, that missing piece right there, more than likely was water. Okay. Water happens to be a very common thing cleaved from a structure. Okay, so our parts, substrate, active site, those are all going to interact via some intermolecular forces. Okay? When we look at our overall reaction, what we now want to do is say, how does this occur? So I'm starting with the enzyme. I'm starting with, or free enzyme. I'm starting with the free substrate. And I'm now trying to make the enzyme substrate complex. Okay. That reaction is going to be in an equilibrium. Just like the substrate could bind to the enzyme and potentially go on to form a product, that substrate could unbind from the enzyme, and we end up right back in the reverse step. So we've got two different steps, or two different reactions that are happening simultaneously through an equilibrium system. Okay. This slide should have shown up in a different location. Some other things that we could potentially look at here, PNP and DADME, or at least that's what I'm calling them. Purine nucleoside phosphorylase metabolizes adenosine into adenine. Okay? So if we take a look, we haven't talked about our nucleotides, but if we look at the left, we've got adenosine, and on the right we have adenine, okay, our nucleobase. So this enzyme comes through, and it cuts and breaks this bond. Okay? That's all it does. It goes through and says, get out of there. Okay? If I now want to build a drug that targets this protein, what might be a potential thing I could build?
Targeted how? To enable it or prevent it?
So usually we're trying to prevent it. Okay, so in this case, let's go simple. We're trying to prevent.
Build a similar key, so to speak, that would bind to the enzyme before the enzyme could bind to the protein.
Exactly right. All we're trying to do is build something that looks like this. Because this is the substrate that goes through and does our end reaction. So if I can build a similar substrate, the enzyme will wrap on that substrate, and it says, well, I can't do that reaction anymore.
But you wouldn't necessarily need to have an entire molecule like that, just part of the molecule
So
bind to part of the enzyme.
how does the enzyme wrap on this molecule? If the enzyme wraps all the way around the molecule, then our target molecule, our drug, whatever we're going to do to try and inhibit, needs to match that structure as close as possible. What if the enzyme only comes up and interacts down here? Then my drug synthesis needs to focus on what structural features are found down there. I could care less about everything else. What if the enzyme binds through this whole section over here?
In biology, we learned like if it binds like that, you would just need to say take up where the nitrogen would lock on, because then the whole enzyme wouldn't be able to fit in there anyway.
All we're looking at is trying to match that site. So what a synthetic chemist might go through and do is build a variety of different molecules. Okay. In this case, and we'll end on this, we've got 6 different molecules here. And notice that they're all fairly similar to our starting structure, but have some pretty key differences. Where do you think our protein likely binds? Those were our targets. What do you think the protein's grabbing a hold of? Well, but if we look at structure 4, structure 4 doesn't have that adenine. And I mean, we go through and look at structure 1, 2, and 3. Those don't really have the adenine molecule either. They do have rings that have nitrogens in them, but it's not adenine. I would guess the enzyme is probably wrapping right there, because that feature is pretty consistent in almost all of those.
Except for 3.
3's got a slight variation there, but we do have that sulfur. We still have the OH. So 3 does have, so maybe
Is it bonding on
it's
the
just
more
the
grabbing here.
2?
Okay. How would we go through and test this? This is my least favorite form of testing. What they go through and do is they build this drug, or all these drugs. They take a 96 well spot plate, okay, with the enzyme, and they put the drugs in each of them and they say, did it work? If it doesn't work, they ignore that drug and they go and look at the one that did work. And then they go through and make a whole bunch of variations off of the structure of the drug in that one, and they do another 96 well plate. Okay. I make this akin to a shotgun method. We take a shotgun, we aim it at a barn, and we find out where it hits. Okay. Even if the shotgun's blast is massive, figure out which one hit, and then we try again. Okay. And we do another shotgun until we try and keep zeroing in on where it actually occurred or where it actually hit, which bullet did best. Okay. Do we know how or why? No. But are we hitting the barn?